We have with us today the freshest and sweetest news from the Korean bio industry. My name is Juri Hwang and I will be delivering some of Korea's hottest biotech issues today for you. So don't forget to subscribe, like, and set your alarm for Bio TV. We'll start off today's bio news on the breaking news from the recent 2 weeks. First, about the partnership between KT and US company NeuroSigma to develop and commercialize electroceuticals at home and abroad. In other news, SK Biopharm has made the great achievement of entering the European market with its anti-epileptic drug cenobamate, while a KAIST research team has developed ultrafast real-time polymerase chain reaction technology that enables rapid thermal cycling and quantitative analysis using plasmonic nanostructures. US bioelectronics developer NeuroSigma now has the first FDA-approved device for treating neuropsychiatric indications. KT, one of the top telecommunications companies in Korea, announced that it has teamed up with NeuroSigma to develop and commercialize electroceuticals to treat neurological and neuropsychological disorders, including ADHD, depression, and epilepsy. The Memorandum of Understanding was signed by the two companies via video conference. Monarch ETNS, developed by NeuroSigma, is the technology that is used to treat neurological and neuropsychological disorders by stimulating trigeminal nerves using an electric patch instead of medications. It has become the first FDA-cleared electroceuticals for treating ADHD. The two companies plan to conduct additional trials for label expansions of this technology for treating other neuropsychiatric disorders such as depression and epilepsy, and to also obtain FDA approval. Existing ADHD medications are typically central nervous system stimulants, which may cause negative side effects such as headache, anxiety, and addiction. However, since ETNS has only minor side effects such as an increase in appetite and a tingling sensation on the skin, it is expected to become a good option for patients and caregivers who are looking for alternatives to those existing medications. KT also established Digital and BioHealth PTF in its Future Value Promotion Office at the end of last year. This year, Kim Young-wook, head of Future Value Promotion Office, was appointed as the chairman of the Digital Health Industry Association, showing the company's great commitment to fostering new healthcare business. SK Biopharm's anti-epileptic drug cenobamate has entered the European market. According to the company, its Italian partner Angelini Pharma has launched the first sales of the novel drug in Germany under the brand name Ontazri in early June. Cenobamate is a novel anti-epileptic drug created solely through SK Biopharm's own technology. It has successfully entered the European market in just 1 year following its first entry into the US market, branded as Exco-Pre last May. Germany, as the largest pharmaceutical market in Europe, is known as the gateway to the wider European market. According to data from Decision Resources Group, the country has 400,000 patients that have epilepsy. SK Biopharm received marketing authorization from the UK Medicines and Healthcare Products Regulatory Agency on the 4th and will soon release the drug to the UK market. The company is expected to collect royalty fees and milestone payments that are linked to its sales performance in the market. In 2019, SK Biopharm signed a deal with Arveo Therapeutics, a Switzerland-based pharmaceutical company, to further develop and commercialize cenobamate in 41 European countries. This January, Arveo Therapeutics was acquired by Italy's major pharmaceutical company, Angelini Pharma. A KAIST research team led by Professor Jung Gi-hoon of the Department of Bio and Brain Engineering announced on the 7th that they had successfully developed ultrafast real-time polymerase chain reaction technology. This technology is known to enable rapid thermal cycling and quantitative analysis using plasmonic nanostructures. These days, the world desperately needs technology that can rapidly and accurately detect infectious viruses to prevent the spread of pandemics, including COVID-19. The standardized COVID-19 RT-PCR testing involves the reverse transcription of RNA, the genetic material of the virus, into the complementary DNA, amplifying the target DNA and detecting the virus with the fluorescent probe. Conventional RT-PCR tests have high sensitivity and accuracy, however, also having the limitations such as a long detection time and the need for samples to be moved to places equipped with large and expensive equipment. The real-time nanoplasmonic PCR developed by the KAIST team combines a vacuum-designed microfluidic chip with a nanoplasmonic substrate with a white light-emitting diode. With this, it is able to rapidly and imply, provide, and quantitative analysis of a small amount of sample and detect viruses in a very short amount of time. This technology is expected to prevent the spread of pathogenic viruses into public areas such as hospitals and curb the inflow of such viruses from overseas. The research team verified the technology using SARS-CoV-2 plasmid DNA and succeeded in quantitative detection with 91% of the target virus amplification efficiency by performing 40 cycles in less than 5 minutes. Since its amplification efficiency is significantly higher and it takes much less time for PCR process to be completed compared to the existing real-time PCR system, it is expected to be suitable for use for on-site diagnosis. Professor Chung said that the team developed ultrafast molecular diagnostics that can be used on-site and that the real-time nanoplasmonic PCR will be a next-generation gene amplification platform for on-site molecular diagnosis. He also stressed that it could contribute to curbing the spread of viruses. Well, that's it for today's news. Don't forget to subscribe to, like, and set your alarm for Bio News. And make sure to have Korea Bio as a friend on Facebook and LinkedIn as well. And I will come back to you in just 2 weeks with more new interesting issues related to the Korean bio industry. Bye.